Good day, this is the Professor, CBMD PhD. And today's Monday, October 28, 2024. It is 9:17 and 14 seconds, p.m. Eastern Standard Time. And this is my lecture on dyes, basic dyes and acidic dyes. So let's on to it here. Basic dyes: methyl green, methylene blue, pyridine G red, toluidine blue. Acidic dyes: acid fuchsin red, aniline blue, eosin red, orange G orange. Anionic components include the phosphate groups of nucleic acids, the sulfate groups of glycosaminoglycans, and the carboxyl groups of proteins. The ability of such anionic groups to react with a basic dye is called basophilia, base-loving. Tissue components that stain with hematoxylin also exhibit basophilia. The reaction of the anionic groups varies with pH. At a high pH about 10, all three groups are ionized and available for reaction by electrostatic linkages with the basic dye. At a slightly acidic to neutral pH, 5 to 7, sulfate and phosphate groups are ionized and available for reaction with the basic dye by electrostatic linkages. At a low pH below 4, only sulfate groups remain ionized and react with basic dyes. Therefore, staining with basic dyes at a specific pH can be used to focus on specific anionic groups because the specific anionic groups are found predominantly on certain macromolecules. The staining serves as an indicator of these macromolecules. As mentioned, hematoxylin is not strictly speaking a basic dye. It is used with a mordant. Important example: an intermediate link between the tissue component and the dye. The mordant causes a stain to resemble a basic dye. The linkage in the tissue mordant hematoxylin complex is not a simple electrostatic linkage. When sections are placed in water, hematoxylin does not dissociate from the tissue. Hematoxylin is best suited to staining sequences in which it is followed by aqueous solutions of acidic dyes. True basic dyes, as distinguished from hematoxylin, are not generally used in sequences in which the basic dye is followed by an acidic dye. True basic dye would dissociate from the tissue during the aqueous washes between the basic and acidic dye solutions. Acidic dyes react with cationic groups in cells and tissues, particularly with the ionized amino groups of proteins. The reaction of cationic groups with an acidic dye is called acidophilia, acid-loving. Reactions of cell and tissue components with acidic dyes are neither as specific nor as precise as reactions with basic dyes. Although electrostatic linkage is a major factor in the primary binding of an acidic dye to the tissue, it is not the only one. Because of this, acidic dyes are sometimes used in combinations to color different tissue constituents selectively. For example, 3 acidic dyes are used in the Mallory staining technique: aniline blue, acidic fuchsin, and orange G. These dyes selectively stain collagen, ordinarily cytoplasm, and red blood cells, respectively. Acidic fuchsin also stains nuclei. In other multiple acidic dye techniques, hematoxylin is used to stain nuclei first, and then acidic dyes are used to stain cytoplasm and extracellular fibers selectively. The selective staining of tissue components by acidic dyes is attributable to relative factors such as the size and degree of aggregation of the dye molecules and the permeability and compactness of the tissue. Basic dyes can also be used in combination or sequentially, e.g., methyl green and pyridine, to study protein synthesis and secretion, but these combinations are not as widely used as acidic dye combinations. And this is Professor Christopher, codename Will, leader of research scientist, ufologist, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover, passion, world peace. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day. Good day, this is the Professor, CBMD PhD. And today's Monday, October 28, 2024. It is 9:17 and 14 seconds.